And left ventricle is a cone-shaped structure with a RVs hugging it. And thickness of the free wall at the base side is about 1.2 to 1.5 centimeter. And it gradually becomes thinner towards the apex. So as Dr. Park mentioned, the apex of the alveoli is very thin, about less than 1 millimeter. It's the same as LV. So even in hypertrophied ventricle has also same thickness of the apex. This is very old literature diagram from the left ventricle. And I'd like to talk in three portions of the left ventricle. First one is outlet, and the second one is muscle itself, and the third one is endocapillary structures. The nomenclature of the anatomy is made by the ex vivo position of the heart. So you need to learn how to, the nomenclature made in the ex vivo is matched to the attitudinal orientation, which means anterior leaflet or right coronary cusp is actually located where in the live body. So actually in the anterior wall. So right coronary cusp is the anterior portion of the aortic aorta. So you need to appreciate the attitudinal orientation of all the structures. So in terms of right ventricle outflow tract, so left ventricular outflow tract is not the left side of the right side heart. So actual position of the left ventricular outflow tract is to the right in LVOT perspective. Left ventricular outflow tract is the region lie between the anterior cusp of the mitral leaflets to the ventricular septum. So it includes perimembranous septum. So membranous septum is a very thin area. You can have a light at the back. You can see that very thin area of the membranous septum. So the outflow tract is very short, and actually there's not much infundibulum, so it's not called infundibular structures. As you know, there's three cusps in the aorta, and the junction with the right and the non-coronary cusp is portion of the membranous septum. So in membranous septum, there's an exact site of the His bundle penetration from the atrium. So it is consistent even in congenital heart disease. So if you look at the His location, it is matched with the junction of non-coronary cusp and the right coronary cusp. So we use the His bundle catheter when do the transeptal. So as a landmark of the lower portion of the aorta. That's the reason here. So the His bundle is kind of insulated with the connective tissues, and it penetrates the membranous septum and divide to the left and the right bundle branches. Aortic root is one of the components of the LVOT. It's a supporting structure of the aortic valve. It's a bridge between the left ventricle and the aorta. So leaflet of aortic valve is kind of semilunar attachment to the aorta and the left ventricle. So it occupied about half of the left muscular wall. So the valve leaflet is kind of wedged to the mitral valve and the tricuspid valve. So the non-coronary cusp is wedged between the two AV valves, and this is left coronary cusp, and this is right coronary cusp. So if you understand this structure, if you map the aortic cusp in the non-coronary cusp, you can see a lot of big aortic atrial signals. That's the reason. So, and another structure is the sinus of Valsalva. Valsalva is between the area between sinotubular junction and the ventricular arterial junctions. So it is most of the sinus of Valsalva is composed by the aortic wall. And at the base of the sinus of Valsalva, there's some sort of ventricular musculature exists because of the semilunar nature of the attachment of aortic valve. And there's not much ventricular muscle in non-coronary cusp and the posterior part of the left coronary cusp because there's an aortomitral continuity. This is fibrous structures, and it's occupied by the left fibrous trigone also. And the semilunar nature of the aortic valve. So aortic valve is attached to aorta and ventricle, semilunar shape, and there's a space between the valve leaflets. So the space between the valve leaflets is called the interleaflet triangle. So interleaflet triangle is about half of them, or sometimes is more than that. And it's bounded by aorta, thin fibrotic wall of the aorta. This is most distal part of the left ventricle, and it can be communicated with the pericardial space or posterior wall of the RV outflow tract. This is a junction between the right and non-coronary cusp, and there's another junction between right and left coronary cusp. This is interleaflet triangles. So there's a typical nature PVC from this interleaflet triangle from the left and right coronary cusp junctions. And another characteristics of the aortic valve is myocardial extension to the aorta. As you know, the anastomosis between the great vessel and the heart is not just clean-cut anastomosis. There's always intermingle of each structures. So the typical example is pulmonary vein with the left atrium. So aorta and the SVC is also had the same histological phenomenon. So ventricular myocardial extension is through the aorta. So most common site is right coronary cusp. And the left coronary cusp and the non-coronary cusp is blocked by the aorta mitral continuity. But it's close to the right cusp, you can see the ventricular extension. So ventricular muscular extension can be found in the interleaflet triangle also. So this is an example of the EP study. It's PVC from the junction between the left and right coronary cusp. So during sinus rhythm, you can see that there's a delay potential at the end of the QRS. This is potential of the ventricular muscular extension to the aorta, and then this signal flipped when the PVCs begin. And this is beginning of the ventricular muscular extension to the ventricle. And this study showed how much ventricular muscular extension exists in the heart. So 95 autopsy cases, and that 22% has ventricular muscular extension. But most of them has in the pulmonary arterial side. So aorta side has about 7% in the study cohort, and most of them exists in the adventitia area. But some of them can extend it to the epicardial side. And I'd like to move to the ventricular muscle itself. As you know, the ventricular muscle has a quite synergistic contraction in terms of working, and that they can made syncytium. And each myocyte is wrapped with an endomysium, which is collagenous connective tissue. It can coordinate the transmission of the force and prevent slippage between cells. And the perimysium is kind of wrapping network of the group of myocyte. It can prevent malalignment between bundles. If there's something changed in connective tissue component or characteristics, it can cause left ventricular dysfunctions. And ventricular muscle, the architecture of the muscle, it is some of you can saw already. There's a kind of sheet of the ventricular muscles. If you can dissect the muscle carefully, you can make a kind of one big rectangular spread sheet. So it cannot be separated by the cleavage plane or sheet, and that there's 3 layers. Superficial layer is about 25% of the wall thickness, very outer side of the layer. It can have oblique direction from the base to the apex. Middle layer is the thickest portion of the muscle, and it has circumferential direction of the muscles. And the deep layer is the thinnest portion, and it has a longitudinal direction from apex to the mitral valve area, and it fused to the trabeculations and continue to the papillary muscle. In hypertrophied muscle, most studies show the deep portion of the muscle can be hypertrophied, and that there's much disorientation of the muscle arrays. And the papillary muscle is the big endocavitary structures in of the left ventricle. And this is a specialized form of trabecular canine, and which is held by trabecular and thread-like bridges. So it's already 66 years, 1966, there's an anatomical and histological study. The papillary muscle is held by trabecular bridge and the kind of fine thread-like bridge. It can hold the papillary muscles. And that bridge contains vascular channels also, quite genius. And the papillary muscle has a big two components, anterolateral area and the posterior medial location. If you think about the mitral leaflet, the anterior leaflet and the posterior leaflet, if you look at the location, attitudinal location of the mitral leaflet, you can assuming where the papillary muscle is located. So base of papillary muscle attached to the middle to apical third of the, with the wide variations person to person. And the anterior lateral leaflet, papillary muscle tend to have single pillar, and the posterior medial papillary muscle has more wide variations. There's a lot of trabeculation towards the apex, and if you look at the base on the papillary muscle, there's not much solid portion base of the papillary muscles. So this is 2004 study with a CT scan. Originally think the papillary muscle is attached to the solid portion of the wall. But if you look at the CT scan with the contrast agent, the base of the papillary muscle, there's a lot of enhancement of contrast agent. So they believe the base of the papillary muscle is seen to attach to the trabeculae, not rather than a solid portion of the wall, which can provide a broader effective base and a multiple point of attachment. So assuming those anatomical attachment to the left ventricle can reduce the stress concentration of the papillary muscle and against the mechanical failure, against the ischemic attack something, yes? And there's a lot of potential for the collateral perfusion of the papillary muscle itself. And then I'd like to move to the electrical system, left bundle branch. As you know, after his bundle penetrates the perimembranous portion, it can become a left bundle branch. It's broad sheath-like structure, and it becomes anterior fascicle and posterior fascicle. But this is 20 normal heart study with a left bundle branch system. So among 20 person, 11 heart has third radiation in the named the median fascicles. So anterior fascicle run through the anterior wall to the anterolateral papillary muscle, and the posterior medial posterior fascicle run through the posterior medial papillary muscle to the free wall, and the septum is innervated by the median fascicles. And the Purkinje fiber. Purkinje fiber is kind of draped the ventricular cavity as a free running fibers. In human study, it penetrates the subendocardium about 1/3 of the myocardial thickness. So each muscular trabeculation has 4 to 8 Purkinje cells. It can isolate with the connective tissues. There's a lot of mesh network with the muscular structure and the Purkinje system. In high magnified view, they found the junction with the Purkinje and the muscular trabeculation. They found that this is kind of electrical signals transmission site, and it's a site of trigger activity of the Purkinje fibers. We are interested in the papillary muscle activation because idiopathic VT include Purkinje network VT called LVOT and the papillary muscle VT. So we are interested in the activation, electrical activation of the papillary muscle. They prepare the canine papillary muscle and look at the activation of the papillary muscle itself with the Purkinje system. So they paste the Purkinje cell. The muscular activation is coming from always from the base. But if they paste the muscle itself, the breakthrough of the Purkinje system is not the base. It's different site. So if we have the papillary muscle VT, the exit to the Purkinje system or the other side can have a different exit or several exits. So this clinical study by the Yamada, the Nikkei group showed that the papillary muscle VT needs sometimes ablation in both sides of the papillary muscles. So this is false tendon. False tendon is cord-like fibromuscular structures across the ventricular cavity. It's not, there's no attachment to the mitral valve, so it's not called tendinate. So among 480 normal heart, 55% of them has a false tendon. But unfortunately, it contains Purkinje fibers. Most of them is related with the papillary muscles. So false tendons are common anatomical normal variants of the human left ventricle, and it can associate clinically PVC and idiopathic LVOT. So anatomic ablation therapy, we need to understand the kind of massive network between the Purkinje system and the muscular trabeculations. So I'd like to move briefly introduce the pericardium. There's not much important structure for the in terms of ablative therapy. So pericardium, as you know, there's 2 layers. So in the anterior side, there's not much obstacle to move the catheters. But if you go to the posterior wall, there's a deflection of the pericardium by some great vessels. So that's called the oblique sinus and the transverse sinus and the pulmonary vein recess, etc. So one thing you need to remember for the pericardial procedure, the phrenic nerve tract. So if when you ablate the pericardial side, epicardial side, you need to keep in mind that the tract of the phrenic nerves. So the epicardial ablation, there's a limitation of the ablative therapy because there's a lack of current shunt by the bloodstream and the lack of convective cooling. And the epicardial fat is quite a very good insulator to deliver the energy to the heart. So there's a study with a conventional RF energy and the cool tip ablation. For the conventional RF on top of the fat, there's no effect. So if you try to burn something in the epicardial side, you need to use the cool tip catheters. So, ladies and gentlemen, in summary, anatomy of the outflow tract and the endocardial endocavitary structure is complex but necessary to appreciate for the electrophysiologist. And the understanding of electrophysiologic phenomenon based on the electrostructural anatomy is crucial to have a good ablation therapy. And understanding the relationship between biophysics of the RF ablation and the epicardial anatomy is important for the effective ablation in epicardial site. Thank you for your attention.